Hello everyone, my name is Dorine Merlat. I'm starting a PhD at the University of Strasbourg in France in collaboration with the Senckenberg Institute in Germany, and I will present my work on annotation and exploration of myriapod genomes. My PhD focuses on some soil animals, the soil being one of the main reservoirs of biodiversity. There is biodiversity as a crucial role in ecosystem as it involved in many processes. However, this biodiversity is managed by human action and their consequences, and it's still largely unknown. This is why the collaborative project MetaInvert was being initiated by the Lowy Center for Translational Bioinformatic Genomics, more simply called TBG. This project aimed to characterize the genomes of soil invertebrates to search for correlations between genomics and ecological traits. For that, the genomes of some invertebrate soil have been sequenced, and among them, 15 belong to myriapods. Myriapods, better known as centipede and millipedes, are small arthropods close to chelicerats and pond crustaceae. And within myriapods, we find subphylum, chilopods, pauropods, and diplopods. To annotate the genomes, we chose to develop our own tools. Because myriapods are non-model species, only few public data are available for the species, and we have many genomes to annotate. Our tool is called EXOGAP for Exotic Organism Genome Annotation Pipeline. It's a snake-make workflow coupling 16 existing programs and 19 scripts I developed. A part of EXOGAP is already available on GitHub, and it is already used to annotate columbola genomes by TBG. In first, programs are automatically installed and configured. Then repetitive elements are annotated and masked in the genome. This is an essential step for the annotation of protein coding genes and for those non-coding. Finally, a last step, the post-processing allows to gather and format the result. And at each step, EXOGAP performs some quality controls. EXOGAP is not yet finalized and some things still need to be improved. For example, we plan to improve the annotation of repetitive elements using additional classification tools, or improve the post-processing step to simplify the deposit in database. And after a benchmarking on model genomes, EXOGAP will be published. We used EXOGAP to annotate the myriapod genomes, but not all assemblies are of sufficient quality to be annotated. We chose to annotate only the more complete genomes. That means the 20 genome with a BUSCO complete score higher than 70%, including 10 chilopod genomes and 10 diplopods genomes. Chilopods are carnivorous and predatory species producing venom. They have only one pair per segment. Diplopods are herbivorous and detrivorous species producing toxin, and they have 2 pairs of legs per segment. And in addition of our 20 genomes, we chose to add 3 published genomes. 1 belongs to chilopods and 2 belongs to diplopods. I will now present some of the results from the annotation, starting with the repetitive elements. We have observed differences in the assembly size, like we can see here, and we wanted to see if these differences could be linked to repetitive elements. And indeed, there is significant correlation between the number of copies of repetitive elements and the assembly size, both in chilopods and diplopods. Repetitive elements are thus key drivers of genome size in myriapods. There is also different types of repetitive elements with a high abundance of LTR, belong to retrotransposon in chilopods, and a high abundance of DNA transposon in diplopods. And these transposons are still active nowadays. Thus, there are different dynamics of repetitive elements between chilopods and diplopods. Here, in orange, you can see the number of predicted protein by EXOGAP for MetaInvert species, and in gray, the number of predicted protein for the 3 published species and for 3 species representing the other arthropod groups, crustaceae, hexapod, and chelicerats. For all non-MetaInvert species, number from common literatures. For myriapods from MetaInvert, between 11 and 16,000 genes are be predicted, which is relatively homogeneous. We also did gene clustering with ortho-MCL to study orthology relationships. This allows us to define the myriapodic pangenome, which represents more than 23,000 genes families. Then we looked at the orthology relationship between the genomes, with in first the gene shared by arthropod, here in red. And we can see they represent more than half of each genomes, indicating a strong conservation between arthropods. For chilopoda-specific genes in green, a few genes only are shared between chilopods and diplopods. In contrast to chilopoda-specific genes in yellow and diplopoda-specific genes in blue, but representing an important part of gene content. And the difference could be the strong differences between these two groups may be explained by their early separation within the myriapods. And to summarize, there is thus a substantial proportion and conserved of gene conserved in arthropod and strong divergence between the two lineages of myriapods. And in the future, we will compare the gene repertoire of the species with the core genomes and the pangenomes for chilopods and diplopods. This will make it possible to outline the gene specific to chilopod, by example relative to venom, and to diplopod, by example relative to toxin. And the pangenome will also be used to clarify the phylogenetic relationships within myriapods and within arthropods. And comparison with other metazoans will allow to analyze the gene involved in segmentation and development of myriapods, and to better define the gene present in the ancestor of arthropods. We will also look for correlation between the genotype and ecological traits, such as habitat adaptation. And according to the Cohen classification, we will compare the gene repertoires present in specialized species, by example, living exclusively in forest and semi-natural areas, and generally species which can live in many environments, to identify the genes and processes related to habitat type for each group. And we'll also focus on detoxification pathway involved in pesticide resistance. To finish, I would like to thank all the people who participated in this work, and thank you for your attention.